The first research project to run on World Community Grid was the Human Proteome Folding Project, calculating the structures of proteins specified by the human genome and other genomes, important to understanding life and disease processes. The project is in its second phase, and so far, the two phases have used almost 150,000 CPU years of computation power, generously donated by World Community Grid members to support the project's research. Please let me introduce Duncan Penfold Brown, who will show you how scientists around the world can access the results of the Human Proteome Folding Project running on World Community Grid.
Hello, my name is Duncan Penfold Brown, and on behalf of the Bonneau Lab of New York University and IBM's World Community Grid project, I would like to talk to you for a minute today about the Human Proteome Folding Project, which is now in its second phase with IBM's World Community Grid. Specifically, I would like to introduce the Human Proteome Folding Project's two publicly available resources, that is web resources, and then walk through an example with these resources focusing on a protein with potential cancer implications in order to show how the general public, or more likely biologists, could mine our data and use our resources to gain scientific insight into important proteins. The first resource I would like to talk about is called the Yeast Resource Center, and it is actually hosted at the University of Washington by collaborators of ours who generously agreed to host and incorporate our data into their system. The Yeast Resource Center, as you'll see on your screen, is composed of many different parts, but we're primarily going to focus on the public data repository, that is the link in the bottom right of your screen. What you're seeing here is effectively the main search interface for the Yeast Resource Center, that is the YRC, and it basically exposes all of the data that we have computed in collaboration with the World Community Grid, as well as data from other sources. Notably, you can see that the Yeast Resource Center contains data on 5,530,000 proteins from many different databases, for which there are about 9.5 million descriptions. That means annotations of some sort. If you look at the changes and additions section of the YRC public data repository, you'll see that one of the last entries is the Human Proteome Folding Project paper. Now, this was a major effort by Kevin Drew and a number of other members of the Bonneau Lab in collaboration with IBM. You'll see a few familiar names down there. And that actually contributed a great deal of data to the YRC. If you look a little further down in the list of changes and additions, you'll see an effort on July 14th of 2008, which is when our project had the first large and major set of data to give to the YRC. That is, we contributed 521,000 predicted domains for 331,000 proteins, which is a massive effort. As you can see there, that data came from 342 distinct organisms. So that's quite a huge set of data that we're working with. One of the challenges with working with such a huge data set is how do you expose that data set to biologists or to any parties that could best use it? And to do that, as we've been talking about, we've developed these two public resources. So the YRC PDR allows you to search for proteins and domains with structure, which is very important for understanding the proteins. You're able to search by keyword, by ID from many different sources, and by sort of general gene name. So that's a very important function for many biologists, but we'll get into the specifics of that later. The second resource I'd like to talk about is one that we've developed in-house, and it is the Proteome Folding Project website, specifically the Yrc Blast Interface. Now, BLAST is sort of an old powerhouse algorithm of bioinformatics that allows one to match a sequence against any number of other sequences. So effectively, it lets you search for a protein sequence against a large number of other sequences. In this case, what we're interested in doing is entering a sequence that we don't know anything about and blasting it or searching it against our database of proteins annotated with structure and functional information. Now, if you're wondering what the use of all this is, why we would want to search for this protein or that protein, why we need to know the structure and function of proteins. And our example will show how you can use basically these two resources that I've just introduced in concert to retrieve very detailed structural and functional information on a certain protein that might indicate to a biologist how that protein works in the body. And that could be very important for example, studying cancer or studying the effects of UV radiation on the human body. Now, I'd like to take a second as well to say that both of these resources are described in detail in the genome research paper that you see on your screen. Again, this is a mammoth effort by Kevin Drew and the Bonneau Lab in collaboration with many others, but especially the World Community Grid. And it was really a landmark effort toward the goal of annotating unknown proteins across many different organisms with three-dimensional structure and then function. Now, let's get into some real-world applications or examples here. As you see on your screen, I have just a simple wiki page up of nucleotide excision repair. So let's say I was a biologist and I was studying the NER, that is nucleotide excision repair, pathways in different organisms. Now, the NER pathway is effectively a process that is responsible for repairing damaged DNA in an organism. So it's basically a group of proteins that floats around an organism and detects damaged DNA. This DNA might run into damage because of UV radiation, which causes mutations in DNA, or other chemicals, or other just anonymous mutations in DNA in general. So what this group of proteins does is it detects that damage, it bonds to the DNA, and it snips out the mutated or damaged sections and replaces those sections with a copy of what the DNA should look like at that point. So it's very important for repairing DNA that's been damaged, especially by UV radiation, which is known in some cases to cause cancer. So, being the diligent biologist that I am, I know that the yeast gene RAD4 is part of the NER pathway in the yeast organism, and I want to know as much as I possibly can about this gene. So I go to the Yrc PDR, as we've seen before, and I search for the gene name RAD4 in a simple keyword search. So hopefully, the results here will show me something about the proteins that make up that gene and maybe their structure, so I can understand physically what those proteins are doing as part of the NER pathway in yeast. So our search for RAD4 has come up with actually a large number of proteins. So we see about 30 here. However, we're mostly interested in the protein structure of proteins that represent the RAD4 gene. So as you'll see, just the top 3 entries there that are annotated in the right column with the PSP, or protein structure prediction keyword. So we're going to refine our search a little bit using that button in the middle of the screen of search results to maybe cut down our results and see exactly what we have here that has protein structure. So the Yrc actually provides a number of sophisticated filters for any search. Of concern to us at the moment is this bottom left box that says experimental data. So as I just said, we're mostly interested in proteins with structural data. So we'll check that box that's also highlighted there that says protein structure prediction. The Yrc also enables you to search by taxonomy or by specific organism. So if we only wanted to search for proteins in human or for proteins in the rice plant, we could do that as well. But for now, let's focus on protein structure prediction. Now we've filtered our search results and we see only 4 proteins, all of which have protein structure prediction data, which is really what we're after. This is actually because structure, or the structure of a protein, as it turns out, is very, very important in terms of understanding what that protein does in an organism. So, as I said, we have these 4 results, and we're interested in the RAD4 protein in yeast. So that is basically the second entry in that list. Saccharomyces is yeast. And then we have 2 following entries, both of which return results for the RAD4 search term, one from a different type of yeast and one from Arabidopsis, which is an important plant organism that is well studied in biology. So let's take a look at that highlighted RAD4 entry for yeast. So here we see the protein overview screen of the Yrc. So we have a description amongst a number of descriptions, and that tells us that we've found exactly what we were looking for. That is, this protein that recognizes and binds damaged DNA, and it's part of the NER pathway. So at the bottom of your screen, you can see the sequence. You can scroll down to see the rest of it. And what follows on the Yrc page for this protein is a great deal of information from other analysis types. So what we're interested in, again, protein structure data, is located near the bottom. So we'll scroll down and take a look. So what you see here is the protein structure data section of the Yrc protein result page. Now this is actually a very dense and seemingly complex section. There's a lot of data on the screen. So I'll do my best to explain it concisely, and if you'd like to read more, please feel free to visit the site and, for example, take a look at that link that says, "What does the above image mean?" So what we have here, the large color block display sort of in the middle of the diagram on top, is a representation of the protein that we are viewing. And the different colors represent different domains. Now, domains are effectively segments of an entire protein that are separate because they are predicted to have independent structure, that is, structure independent from the other domains of a protein. And this RAD4 protein that we're viewing now, we have broken up into 3 different domains. That is, our methods have broken them up. Now, unfortunately, as you can see in the bottom section of your screen where each domain is described, there are no confident structure predictions available for any of the domains. This occurs at times because the domains themselves may actually be too complex for our very state-of-the-art software to predict. This is not at all uncommon. It's a very difficult problem. However, just for fun, we are going to take a look at these domains and try to see if we can find any structure to look at, just so you can see how the website works. We'll get to some confident structure predictions in a second. So simply click on the View Details of any domain in that list, and you will be shown potential structures. So here we are with the display of the details of each predicted domain. As we said, there were no confident structure predictions for each domain, but just to see, let's click on this highlighted link here that says click to view all matches, which will show you all predictions, whether confident or not. Now, having clicked that link, we see that predicted domains 1 and 2 still have no structures, but the third predicted domain actually has a large number of them, some of which have a fair probability, or MCM score. So, for example, that which is highlighted on your screen has an MCM score or probability of 0.772, and that's not bad. So if we want to take a look at this structure, because again, we're interested in this RAD4 protein, and we want to see how it actually exists in the yeast organism, we can take a look by clicking on that view link, which will open a pop-up, or we can simply download the structure file to view on our own computer with our own software. Note too that if you consider the domain diagram of this protein, that the third predicted domain is just this little tiny piece shown in magenta at the end, as we saw a few pictures back. So we're looking at the structure here of a very small tail piece of this protein. So clicking on that view link will result in a pop-up window with an embedded JMOL applet. Now this applet, as you can see, will allow you to view the predicted structure of this domain in 3-dimensional space. So there you see we have this little wiggly protein that is composed basically of 3 very small alpha helices. These are those wrapped magenta structures you see, linked together by some sort of non-structured or loosely structured coils. Now this can be very cool for a biologist, or especially a structural chemist, for example, because they can move around this protein in 3-dimensional space and see how it's actually shaped, theoretically, in the yeast organism. And that can actually give many clues as to what it does and how it functions. Again, being that this is only the tail segment of a fairly large protein, this simply gives us a clue of what the very end of that whole protein might look like, which is actually of great effect for some biologists because it might determine how the protein binds to something at its end, or initially discovers damaged DNA in this case. Now, as mentioned, for this protein RAD4, we had no very confident structure predictions, even though that last one was not bad. But even that last one was just a tiny segment of the protein. So let's return to the search results and see if we can find anything more promising in terms of a structural representation of the RAD4 gene or protein. So back to our search results, let's, out of curiosity, take a look at this RAD4 protein from Arabidopsis. And one of the cool things about this data set and this resource, which allows you to search amongst it, is that it will show you information from many, many different organisms. And that can be interesting just in terms of considering how protein function or gene function is conserved across organisms. So, as I said, let's take a look at this Arabidopsis protein. So looking at the protein structure data of our newly selected protein, we can see again in this large color block representation that the protein has been split into 3 domains. However, this time the center domain, or domain 2, is large and dominates the protein and is matched with a very confident method to a known structure. So let's take a look at that structure, review details of the domain. Now, looking at the details of this predicted domain 2, we see that the detection method is Pfp Blast, which is with a very high confidence score, to the matching structure 2QSFA, which is a PDB structure. The PDB is simply the Protein Data Bank, which is a resource in which biologists store many known structures, or many structures that are proven to exist. So let's take a look at that by clicking on the 2QSFA link and checking out the PDB. So the Yrc will link to the PDB structure, which will open in a new window. And I'm not going to go into much detail about this site specifically, except as said, the PDB, or Protein Data Bank, is simply a resource for biologists or people in other disciplines to store data on known protein structures, that is, protein structures that they have experimentally verified. So here we basically are seeing information that that central domain of our RAD4 protein from Arabidopsis has this structure at a very high confidence. So that's actually great, because through this site we're able to read about the structure, to see what other scientists have found out about it, and to look at its structure again in 3-dimensional space to make our own conclusions about it. Now, we've seen how the Yrc PDR can help us understand information about a protein, especially structural information. We simply have to visit the Yrc site, search for the information we're looking for, and examine the results that come up. So I would like to introduce this second resource, and to do that, I'll motivate it through trying to use the Yrc first. So, again, the diligent biologist, I know that while RAD4 is a gene that's part of the NER pathway of yeast, I also know that this RAD4 gene has a very similar gene called XPC, which does the same thing as RAD4, except in human and other mammal organisms. So I'm going to search for this XPC gene on the Yrc, as you see. So, searching for XPC, as we did for RAD4, we come up with a number of proteins. We have 12 proteins here, only the first of which has protein structure prediction data, and we see that that protein is in fact the same RAD4 protein we viewed in the first example. So that's no good. We've already looked at that. So it seems that the Yrc does not have any information on the proteins that make up this XPC gene. So what can we do here? And this is what motivates the second resource. As previously mentioned, the in-house developed Pfp Blast Interface. So, as mentioned, the Proteome Folding Project, or Pfp Blast Interface, allows you to enter a sequence, protein sequence of your choice, and it then attempts to match that protein sequence against protein sequences that we have already analyzed. That is, protein sequences that may have structural prediction data. So I happen to have a protein sequence that represents the XPC gene in human. As Homo sapiens, you see it as I've entered it in the text box in the Blast interface. So I'm just going to do that. I'm going to drop the sequence into that box and enter my email address. and choose at that box at the bottom the PSSP database to blast against, which is protein sequences with structure predictions. So hopefully when we get the results of this blast sequence search, we'll get some telling results that give us some information about how the XPC gene and proteins function in the NER pathways in humans and other mammals. So as you can see, once you submit a blast sequence search, you're given a status page that will automatically update, and when the job is finished, this field successful will be true, and you'll also be sent an email that will link to your results. So once you have been notified that the sequence search is completed, you can scroll down on the results page or follow the link in your email to see the blast high-scoring pairs, or the blast results that have matched well to the sequence you put in. Quickly, to explain these results, you see here we have a number of results, maybe 15 or so. The expect column in the left-hand value you can think of as a score where the lower number is better. So the first score being 0 means we have an exact or a near exact match. Now this alignment box next to the expect value, over to the right, shows the entire space of the protein that you entered. So if you can look at the second result and see that the white parts of that box are the parts of the sequence you entered that do not match to the entry that is showing you right there. So again, in the first example, or for the first result, the box being all blue indicates that the entire protein is matched. Now let's take a look at some of these sequence search results. The first result, as we said, matches the entire protein. And it should be mentioned here that the entire purpose of this blast interface is to link to proteins that are in the Yrc database that we had been examining earlier. So as we see for the first result, there is an exact match in this database. However, no records are found in the Yrc, so we have nothing to link to. Notably, however, on the bottom of your screen you can see that you are able to view the query, which is the sequence that you submitted, and the subject, which is the sequence that's being matched to in our database, and you can see how the two are aligned. So you'll notice that the alignment is nearly perfect with the addition of some gaps in the sequence that we submitted. So let's take a look at the second match with an expect value that is, as you can see, very, very low. It's highlighted there, low being very, very good. So in this case, we have a high-scoring match with a number of links to proteins that are in the Yrc. So you see those in the list that takes up the bottom half of your screen. In this list, we notice a lot of results from Drosophila, which is a fly, sort of an interesting result, but it's based on sequence similarity, so we have to have some kind of traction here. What I've highlighted near the top of the screen is the query start and end and the subject start and end, and these are the ranges that this match occurred at. So it's notable because the sequence that we searched against this database matched all of these fly proteins from the range of 923 to 1293, so quite far to the right or far along in the protein. So keep that in mind when we view these proteins on the Yrc, which we can do by clicking on the links at the left. So by clicking on the links of the sequence search results that we got from the Pfp Blast resource will lead us directly to a Yrc protein overview page, as we've seen before. So here is the protein overview page for one of the fly proteins that matched to our human XPC sequence. Let's take a look at the protein structure data of this protein and see if we can figure out why it is that our human XPC protein sequence, which is part of the NER pathway in human, matched to this fly protein. Now keep in mind that the human XPC protein sequence that we used to search our database with the Pfp Blast Interface matched to a protein that we're currently viewing, but matched specifically to the region from somewhere around 900 residues to 1290 residues roughly. So, as we're already familiar with, this is the protein structure data section of the Yrc, and we can see that the protein that we've matched to is broken up into a number of domains. And notably, the human XPC protein that is part of the NER pathway in human that we use to get this matching result matches to the ladder domain of this protein you see on your screen. That is, the orange domain, domain 6, takes up the entire space that our match sort of found. Importantly, as you see highlighted there, this orange domain is assigned to the structure 2QSH, which, if you'll recall, is the same structure that the Arabidopsis Rad4 protein earlier also matched to. Overall, this tells us that many of the proteins that are implicated in the nucleotide excision repair pathway, whether they're from yeast or Arabidopsis, a plant, or human or fly, exhibit the same structures, which is very interesting because it says that, first of all, structure is conserved across organism when a specific function is needed. It also gives us a lot of specific information about how nucleotide excision repair is initiated based on this one protein structure, and that's very valuable for some biologists. So that wraps up our practical example. It's pretty spread out and definitely a mouthful, but that's sort of what we deal with all the time, and that's what these two resources make available to us. Again, we have the public data repository, which you see here from the Yeast Resource Center at the University of Washington, which we can use to view protein and protein structure data by searching for gene names or protein IDs, and the Proteome folding project Yrc Blast Interface, which effectively lets us search for protein sequences against a database annotated with protein structure, which links directly back to the YRC for display. Again, my name is Duncan Penfold Brown, and thanks for listening. Also, a big thank you to IBM's World Community Grid for providing us with countless hours of CPU time, without which we would have been able to do none of this interesting science. So, thank you to the organization, and especially to the users.